So pines are conservative water users. We see that even under a mild water deficit, they will close their stomata. So that means they're not transpiring, and we would predict then that because they're closing their stomata, that photosynthesis would be reduced, and that's going to reduce the overall carbon gain. So that's the carbon that's available for growth or partitioning into defensive pathways. And this is in fact what we see then. This is one of 6 independent biological replicates that we have done over space and time with different provenances of seedlings. And so we get this pattern 5 out of 6 times where both lodgepole pine and jack pine show this reduced photosynthesis fairly early on in terms of the effect of water deficit versus well-watered trees. But in these early stages, we don't see an effect of the pathogen on reducing photosynthesis. So the tree is coping at this point. If we extend this experiment way, way, way out, we can see a small effect of the pathogen on photosynthesis, but that's also confounded with the conclusion of the growing season. It's likely that the pathogen-affected trees are simply moving into dormancy more quickly. So where the rubber hits the road then is knowing whether that mild water deficit can affect the tree's defense response. So we've used lesion development, as many people have, as a measure of the tree's defense response to this attack. And what we see is that lodgepole pine mounts a very rapid attack against inoculation by the Grossmannia clavidra, and presumably then contains that infection, because we see that this discoloration or this deposition of secondary metabolite then subsides in later days. Under water deficit, this response is slowed. And this is really important. If we go back to the classic plant pathology literature of all the work that's been carried out in crop plants, we know that the more rapidly a plant is able to detect that it's being attacked and mount that defense response, it contains that pathogen. And the pathogen does not win the arms race. The slower that response, the slower the plant is able to perceive that it's been attacked, and the slower that it mounts that defense response, the more quickly or the more easily that pathogen can establish and win that race. So we see that jack pine then has a much slower response. And so this is consistent with the idea that jack pine is not a co-evolved host with this fungus, and so it may not have that early surveillance mechanism. And our patterns overall then agree with the mature tree patterns. And recently, Adriana, who's done just an amazing job on this project, has developed from our genomic resources the ability to detect where the fungus has grown within inside this lesion, because the question is, well, we're looking at a response of the tree and we're inferring that this is a containment, but where is that fungus really growing, and did that defense response really effectively contain that fungus? And so now with those genomic tools, we can go in and say, well, did the fungus get to here? Did the fungus get to here? Did it get to here? Or maybe it escaped altogether. And so that will allow us to get at one of the more contentious issues in mountain pine beetle work, and that is, what does the lesion really tell us? And so we'll be able to, we also live in next year country, and so next year we'll be able to tell you what that really means. So we've also looked at the expression of a whole bunch of genes associated with water limitation responses as well as with defense responses. I'm going to just show you a small number here. These two genes are chitinases. These are part of the plant's nitrogen-based defense responses. So chitinases are enzymes that chew up fungal cell walls. We know from the classification of these chitinases that they're likely classic chitinases. These two genes are involved in terpenoid biosynthesis, terpenoids being part of that pitch and the volatiles that are produced as part of that defense response. These have been well studied then in the conifer system. And so what we see then is that for some of these genes, we get an induced or an increased expression constitutively. So the preformed responses under water deficit are actually enhanced. But what we see then also with some of the other genes is that in the induced response, so once the plant is inoculated, that the plant is unable then to mount an induced response of some of these genes. Not all of the genes follow the same pattern. So we don't have a wholesale increase in the constitutive defense response. We don't have a wholesale decrease in the induced response. We have selective changes in certain parts of that defense strategy that may overall compromise that plant's response. So maybe under mild water deficit, there's an initial barrier to successful attack. But if that initial barrier is overcome, the plant is spent. There's just not enough, as we heard yesterday, the analogy money in the bank, that can be spent on additional defense responses. So these are 4 genes of the 20 that we've looked at. And in our next year country, we currently are analyzing a whole suite of genes using these 25,000 gene microarrays. We've just completed 168 arrays that Adriana is madly trying to process. Each one of these slides then presents about 80 gigs worth of data, so you can imagine that this is a pretty big deal. We have another 186 on the way to look at what the responses are in the wood, because we know that if we just look at the bark, we know that that's where the initial defense response is happening, but we know that there's core communication going on with the wood as well. And so we're looking at, because the wood is where the water limitation is really sensed, we're looking at that communication and how that water deficit that is perceived in the wood might be affecting the phloem as well. So these will tell us genes then that whose expression responds to differences between the species or differences in the environmental treatment that may be important to the plant's response to attack. But ultimately what we really want to know is that there's genetic variation across the landscape. Do we see heterogeneity across the landscape in terms of the tree's ability to respond? So for example, one might hypothesize that in the drier regions of Alberta, we heard mentioned yesterday that Alberta presents a drier environment than BC, and so water limitation is an issue here. For example, are those trees more adapted because they're living in a more water-limited environment to cope with that chronic water limitation, and maybe their defense strategies are not as compromised as those trees that may be in the more mesic environments that are subjected to episodic events of water limitation. And so we can look at that adaptive variation over the landscape by choosing these candidate genes that come out of the gene expression analyses and looking at the variation in their coding sequence across the landscape. And that's where the collaboration between Adriana and Kathy is really important. We designed a chip that has over 1,000 different markers on it. This is work that's progressing day by day, so Kathy on Monday is going to give us an update on this to look at what kind of signatures of selection may exist in the landscape to say these trees over here, they're different than these trees over here. And that might come into play, again, in the adaptive forest management type of framework or strategy, where an understanding of what that genetic variation looks like on the landscape could be used maybe to inform seed zone strategies, or reforestation strategies, or understanding really what the biodiversity of individuals on the landscape are. And so here we have just one example of genetic variation for one single gene plotted on an overlay of seed zones that the province has devised. And you can see that there's some need agreement there. And there's also some disagreement there. And maybe some of these seed zones, in this particular instance, could be combined, or maybe it shows where splits might need to be. If we're thinking about forests in the future, are these seed zones that we think about today, are they going to be applicable in 100 or 200 or 1,000 years' time? So we can use genetic variation then and use the marker data that Kathy's developing to work with reforestation planning to maybe infuse some genetics into reforestation planning, particularly into seed zones. This is a really exciting area of work, I think, and something that we'd like to pursue in more detail. So we're using similar types of approaches to look at genetic variation in this beastie, mountain pine beetle. And I'm just going to, because this is work that's going to be described in a lot more detail on Monday, I'm just going to hit the highlights. We have one sex-linked genetic marker that we can use then to examine sex ratios across our collections. So for those of you who haven't done sex typing of beetles, I haven't done it myself, but I shared a printer with somebody who was really enthralled by it, and I think it's actually quite laborious. So close your eyes and just imagine sex typing 2,500 beetles, because that's what this sample represents. And so you can see across the landscape, that ratio of male to female is not the same. What we have here actually are the leading edge populations that were sampled in 2007, and we can see those ratios are quite different than, say, some of these ratios in more established areas that were collected a little bit earlier. We have now replicated samples from these same sites that were taken in 2010, 2011. It would be interesting to see whether those sex ratios changed. And so with these markers, one thing I'd like to mention is that with the new funding that we've received from Alberta Innovates, we're going to be making these markers available as a fee-for-service, where it's essentially a full-service genotyping facility that's working within our department, where you can supply the samples, we'll supply the know-how, we'll be able to generate reports with them that will be interpretable by non-specialists so that you'll get the data that you need. We'll work together with you. So these sex-linked markers, together with the species discriminating markers, are now being made available to interested end users. So if you are interested in them, please don't hesitate to drop us a line. So the sex-linked marker was not included in this analysis, but we used a number of other genetic markers to look at population structures of mountain pine beetle across the outbreak in Western Canada. These are the Herculean efforts of ASRD and our beetle collecting crew that has gained some infamy in time. And so using those genetic markers, we can see that we can differentiate between the northern populations and the southern populations. These northern populations then showing classical signatures of expansion, which ties into this the holy grail of dispersal biology of insects. We also can see that there's strong genetic connectivity between these populations of northern British Columbia and these founder populations in northern Alberta. So strengthening the supposition that these beetles arrived from northern British Columbia, where they arrived from. We wouldn't be able to distinguish that because these guys are all basically genetically very similar. But on the whole scale of things, if you look at these data, they don't show a great amount of something that we call isolation by distance. So if I take this group of beetles and compare it to this group of beetles, they're not very genetically distinguishable. So what that means is that, as we all know, beetles can fly long distances. And so we don't get these little pockets of differentiation the way that we might or expect to see in other systems. And so that's telling us something about the biology of the system. But despite that, we know that we have huge selective pressures, particularly on those northern populations that are moving into novel habitats. And one of those huge selective pressures then are the winter cold temperatures. So we can expect that, and we know from the work that the modelers have done, that ASRD has done, that when beetles fly into an area and they overwinter, lots and lots and lots of them are killed. Although in the mortal words of Dan Lux, lots minus lots is still lots. But say we've got this guy. What do we call him, Kathy? Mr. Super Freeze?
Mr. Freeze.
Mr. Freeze. So Mr. Freeze here, maybe he's got a genetic background that enables him to attain cold hardiness faster, maybe to withstand colder temperatures for longer. If we have a huge amount of winter kills, say in the 80+% overwintering mortality, what we might see is that Mr. Freeze is going to survive, but a lot of his brethren aren't. So we've selected for the alleles carried by Mr. Freeze. And so if we're getting this kind of selective pressure every single winter, even in mild winters like we've still had quite a substantial selective pressure relative to the types of selective pressures that you might see on other types of organisms. It won't take very long before Mr. Freeze is a popular guy in his bunch, and maybe has gone out and been promiscuous and made all sorts of little freezes. So we can imagine that we've got a situation of rapid adaptation happening. So we are currently using this marker technology to see if we can find these signatures of selection that may be saying that we're capturing adaptation in real time. And so Jasmine, who's not with us today, is sitting under a veritable mountain of data, determining whether she can detect these signatures of selection, particularly for genes that we hypothesize are involved in cold tolerance. And that's work that's being done in collaboration then with Dazine Huber, who's at University of Northern BC. So here's the teaser. A number of the different parameters and insights that we've gained from the genomic studies that I've talked about at this point can really inform the modeling process, whether it's through resinosis or dispersal, selection, or adaptation. We didn't talk about nutrition, but we have data on that as well. The notion of overwintering. These data might be able to feed directly into models, or they might be able to say these parameters are important in the modeling process. And so on Monday, Barry's going to give you some insights on how these data have been used so far. And he'll also tell us something about this crazy diagram, which says that sometimes if you're trying to develop risk models, sometimes the risk assessment process itself is as important. And so we've been able to use insights from the project to date, such as the finding that mountain pine beetle had established on what we could genetically determine to be jack pine, to inform the risk assessment process. And so, especially through the National Forest Best strategy, this information was used at a national level to really highlight the importance of the work that's been done here by SRD and spread control, and to get the buy-in of provinces east of here that spread control really did matter, because what had been perceived as a barrier was not in fact a barrier. So just to summarize, because I know I'm way over time now, this outbreak has given us an opportunity to do the proof of concept that you can take approaches like genomics that have been used very successfully in medicine and apply them in an ecological context. But it takes coordination and collaboration with people who know the system and to get out and do the ecology. And that's, again, I just want to mention that this ties in with work that Maya's going to talk about after the break. What we can take home is that pine and mountain pine beetle populations are heterogeneous. And so this really has impacts for modeling. We can't assume that this is like an oilcloth tablecloth where if you spill a glass of milk, that milk's just going to flow uniformly over that landscape. There's going to be barriers there, but how big are those barriers? Are they significant? Are they insignificant? Under what conditions do they become important for the beetle? And most importantly, if the beetle does move through an area and there is large-scale mortality, what do you do afterwards? What about the reforestation? So Barry, as the teaser, we'll talk about on Monday how we've used genomics to inform the risk assessment process as well as risk modeling. And we think that some of the genomic insights that we have so far can be used in reforestation as well as in genetic conservation strategies and in policy development. So with that, I really want to thank the people that I'm up here talking for, particularly Adrienne and Kathy, who have done just an amazing job with this project, my co-investigators on the material that I've talked about today, and a whole bunch of other people who made this project happen. And I especially want to acknowledge the role of ASRD, not only as funding partners for the TRIA project, but really playing such an instrumental role, particularly in the early stage of the project. And I especially want to thank Don Harrison, who obviously is not in the room, but I want to thank him for his insights, his early insights early on in the project, his championing of the project, but also his years of experience in the 80s outbreak were really instrumental in getting us off the ground. So I do want to thank him as well.